Morning, everyone. Good morning. Can you hear me? My screen share is still disabled.
Morning, doctor.
Okay. Good morning. Now it's enabled. Thank you.
Yes, doctor, now you're a co-host. You can start sharing your screen.
Okay. Thank you. Thank you so much.
Thank you, doctor.
So good morning, everyone. Today we are going to be discussing food poisoning and Helicobacter pylori. I'm going to start off with food poisoning. And these are the learning objectives. By the end of my discussion with you today, you should be able to describe the bacteriology in terms of the common agents of food poisoning and their characteristics. What are the risk factors and what is clinical presentation like? You should be able to discuss the principle of management and laboratory approaches to the diagnosis. So this is my outline. Let's start. Food poisoning is a common condition worldwide. It may present with self-limiting diarrhea, or you may have diarrhea with associated vomiting. Most of the time, the basic mechanism responsible for food poisoning is the ingestion of preformed bacterial toxins. These may be exotoxins or enterotoxins, which are already present in the food. Examples include Staph aureus enterotoxin and Bacillus cereus emetic toxin, that is, it causes vomiting. At times, food poisoning may be as a result of the ingestion of spores, not really the toxins. In this case, the spores will get to the GI and then convert to the vegetative form of the organism and start producing toxins. So a good example is Clostridium botulinum. Now, the hallmark of food poisoning is that you're going to have a lot of patients presenting to the clinic with similar symptoms. And if you take a very good history, you find out that there is a common source of exposure. What do I mean by this? Patient A comes in. He attended his friend's convocation party the day before. While at the party, he had a particular dish, okay? Then patient B, C, D, E came in. Similar complaint, similar history of having attended a convocation party the day before. So when you have a lot of people with similar symptoms and you take a food history, you find that there is a common denominator, either because of the dish or the event that they attended, then you should be thinking of food poisoning. So what are the risk factors? After all, we are dealing with cooked food, right? We are not eating the food raw. So what happens is this. We have, sorry, I'm trying to get a pointer. Can't seem to see it. Oh dear. Okay. Can't find my pointer icon. Okay, if you can see the mouse. So we have a food handler who is colonized. That person is regarded as the source of contamination. Then you have prepared food. So you are not likely to cook all over again. At best, the food is going to be reheated, but it's not going to be cooked all over again. So this person handles the food and in the process transfers these bacterial agents onto the food source. Now the food is kept at a particular temperature for a certain period of time. Now the temperature is such that It supports the rapid growth of bacteria. So the bacteria is going to metabolize quickly and start producing toxin. Now, what temperature range is regarded as the danger zone? It's usually the temperature between 5 degrees and 60 degrees centigrade. Because below 5 degrees, you have the cold and the frozen food zone. At this temperature, bacterial metabolism is slowed down. The temperature does not really support bacterial metabolism, so they don't grow, they won't be producing toxins. At the hot food zone, which is above 60 degrees, the organisms are actually destroyed, so there's nothing to produce toxins. However, between the temperature 5 degrees and 60 degrees, which is what you have here, because when you go for the party, the food will be on the table for some time. You'll have some fun before you start eating. So that's granting the bacteria enough time to replicate and start producing toxin. Then when you're done with all the fun, you start to eat. The toxins are already present in the food, and eventually the patient comes down with food poisoning. Now let's discuss Staph aureus food poisoning. Symptoms usually begin within 1 to 6 hours. What I want you to note is that with this condition, there is no fever. The moment the patient has fever, you are most likely not dealing with food poisoning. You probably have an invasive or infective process going on. Now, the organism has 22 serologically distinct enterotoxins, and we actually use these enterotoxins to characterize Staph aureus when we perform molecular assays. There are different forms of enterotoxins. You have enterotoxins A to E, which are regarded as heat-stable toxins. Now, food products that have a high protein content are usually associated with Staph aureus food poisoning. So what do you have? The food contains protein. It is cooked, like I said earlier. But then you have a colonized food handler who contaminates the food after the cooking process. Now the cooking process gets rid of competing bacterial agents. Then this contaminated food handler introduces new organisms that are going to thrive. Room temperature is within the temperature zone, the danger temperature zone. And then the organism is going to incubate, the organism is going to metabolize the food substrate, and then it starts to elaborate toxins. Now note that because these toxins are stable, reheating the food will not eliminate the toxin. It may kill the organism, but the toxins are still there. So after consumption, the patient becomes symptomatic within 1 to 6 hours. Now, the mechanism of action of the enterotoxins are not completely understood, but it is believed that they act as super antigens. So what is the laboratory approach like? Because this is a toxin-mediated condition, you don't need to culture the organism from stool. So diagnosis is purely clinical, and you must take a very good food history. However, if you really want to make a cultural diagnosis, you want to isolate the organism, you want to screen for the type of toxin, your best samples to send to the lab is the food remnant or whatever the patient threw up. This is what is sent to the lab. And you still remember the laboratory features of Staph aureus. They are gram-positive cocci in clusters. They're catalase and coagulase positive. And then we go ahead to perform molecular assays to screen for the toxins. Another agent that is commonly associated with food poisoning is Bacillus cereus. Unlike Staph aureus, toxin production is associated with starchy food, particularly rice, white rice, fried rice. Now, there are associated distinct clinical forms of food poisoning due to this organism. You can have the one that is characterized by predominantly vomiting, that's the emetic syndrome, and you have the other which is characterized majorly by diarrheal illness. So the emetic syndrome is due to a highly stable toxin, stable in the sense that it is resistant to high temperature, it is resistant to enzyme digestion, and extreme pH. While the diarrheal form is mediated by a toxin that is heat and pH labile and can be degraded by enzymes. The toxins, just like with Staph aureus, may be screened for in food remnants and vomiters. So this table is contrasting the clinical forms of B-series food poisoning in terms of the implicated food, the incubation period, the symptoms, the duration from exposure to the first clinical symptoms, and the form of enterotoxin that is involved. Now, with the emetic form, which is characterized most significantly by vomiting, the food implicated is rice. Incubation period is usually less than 6 hours with a mean of 2 hours. The toxin involved is heat stable and it takes about 8 to 10 hours for the disease to last out. These conditions are self-limiting. How do you make a diagnosis? Like we said, toxin screening in the vomiters or the food remnants. However, for public health significance, there are times when you need to isolate the organism from maybe the food. So what are the laboratory features of this organism when you isolate them? They are catalase positive. They are gram-positive rods, as you can see in the micrograph here. They are spore-forming and they are motile. And because they can produce toxins, we can also perform a toxin screen in the lab. Another organism you should know about is Clostridium botulinum. This is a very dangerous organism, and the botulinum toxin is a very potent toxin. The disease condition associated with Clostridium botulinum is called botulism. It is rare, luckily, but it is potentially life-threatening because it is a neuroparalytic effect that the toxin has on an exposed individual. Now, the normal actions that you have at the neuromuscular junction is that when excitatory signals from the central nervous system get to the muscles, acetylcholine is required to effect the contraction of muscle fibers. The botulinum toxin inhibits the release of the acetylcholine, thus inhibiting contraction. So when you have botulism, you can have progressive paralysis that may eventually involve the respiratory muscles. As a result, it is a life-threatening condition. What are the risk factors? What can predispose a patient to the toxins? Consumption of home canned food or homemade honey are the leading risk factors associated with exposure to this organism. The symptoms may begin within 18 to 36 hours after ingestion of contaminated food. Usually, the spores are ingested. The vegetative organism will be destroyed by the gastric acid. So the spores will survive the transit through the stomach, and then in the small intestine and the large intestine, they become vegetative organisms that start to produce the toxins. The symptoms include double or blurred vision. In young children, you may notice that they have drooping eyelids, slurred speech. There may be difficulty in swallowing or sucking breast. There may be dry mouth and muscle weakness. And like I said earlier, respiratory muscle weakness may lead to death. Now, there are different types of botulism. We are mainly interested in the food-borne botulism, but I think you should be aware of the other forms. The iatrogenic botulism is due to cosmetic application of Botox. At times, you may have an overdose of this injection, and you could have the symptoms I've highlighted in the previous slides. Now, with infant botulism, it's usually as a result of feeding children below the age of 1 with honey, particularly homemade honey. And this is one of the reasons why we don't encourage children below the age of 1 to be fed honey, because you don't know if the spores of these organisms this organism is present in the honey, and you know what can happen when the infant is exposed. Now, in terms of management of botulism, it's usually supportive care, particularly when there's respiratory compromise. So you need to intervene until the effect of the toxin wears off. You can also provide the patient with antitoxin. Now, management of food poisoning in general. Note that food poisoning is a self-limiting condition because the body gets rid of the toxin with time. The vomiting, the stooling is all aimed at getting rid of the toxin. And once the toxin is out, it's out, so the patient will recover. In terms of patient presentation, take a very good food history. You've been to a party recently. Oh, what did you eat? How many of you came down? Are you aware? Things like that. So you must take a very good food history. In the elderly, you may need to intervene by giving intravenous fluid. The use of antibiotics is controversial, but depending on the patient's immunocompetency, you may need to use antibiotics at times. But other than that, in an immunocompetent person, fluid replacement therapy is all that is needed. Now, the patient should be educated to take preventive measures. For example, patients who can their own vegetables at home should observe this food for bulging, because that suggests that you have some form of metabolism, microbial metabolism going on with gas production. That's why you have that bulging. Hands should be washed before preparing food and before eating food. Store cooked food at the appropriate temperature. And the best option is to always eat your food as soon as possible after cooking. So this is a more extensive list of what you should do to prevent food poisoning, and it is self-explanatory. So in conclusion, food poisoning, it's usually a self-limiting condition, and your diagnosis is clinical most of the time. It's mediated by toxins, so you need the presence of the toxin to make a diagnosis. You don't need to culture the organism from the patient's stool. Food history is very important, and supportive care may be needed in some cases. So this ends food poisoning. Before I start with Helicobacter pylori, do you have any questions? Okay. So let's move on to Helicobacter pylori. By the end of this discussion, you should be able to discuss the morphology and the microbiological characteristics of Helicobacter pylori. List the various virulence factors because this is what is important in the disease process. So you should be able to list the various virulence factors associated with the organism and its pathogenic effect. How is the organism transmitted and the laboratory diagnosis? So that's my outline. Before the discovery and isolation of this organism, it was believed that peptic ulcer disease was due to stress or taking spicy foods. However, these two gentlemen were able to definitively isolate and identify and characterize this organism in 1982. So what they did was that it's good to have good friends. Robin and Barry were friends. One is a pathologist, the other is an internal medicine physician. So he believed that peptic ulcer disease is due to a microbial agent, but he couldn't isolate the organism. Then he had to go on a break. He forgot his culture plates, and then when he came back, he saw the beautiful growth. What he did was to drink from the culture broth he prepared from patients with peptic ulcer, and he came down with symptoms of peptic ulcer disease. His friend took biopsies, and they were able to conclusively demonstrate the presence of the organism in the biopsied tissue samples. So the two of them received the Nobel Prize for Medicine in 2005 for this study. Helicobacter pylori is recognized as a type 1 carcinogen. What that means is that this organism is directly linked to cancer formation. It is so designated by the International Agency for the Research on Cancer, IARC, and the World Health Organization. Approximately half of the world's population is believed to be infected by the organism. It is believed that most people are exposed at a very young age. It is asymptomatic in most patients. Long-term carriage, however, is associated with increased risk of site-specific disease. For example, about 10% of people with long-term carriage eventually develop peptic ulcer disease. About 1 to 3% will develop gastric adenocarcinoma, and less than 0.1% end up developing mucosa-associated lymphoid tissue lymphoma. Like I said, it has a worldwide occurrence. About half of the world population is believed to be exposed. There could be variable colonization rates. For example, in areas where you have poor hygiene, particularly food, environmental hygiene, water hygiene, there seems to be a higher colonization rate than in developed countries where you have better sanitation, better provision of pipe-borne water and things like that. Even in the same country, there is a variation in the colonization rate among different population groups. So in the developing country, you can have the elites. There is variable colonization rate compared to the less privileged in that particular environment. So the very rich, in quotes, in developing countries have a relatively lower colonization rate when you compare them to the less rich in the same country. And like I said earlier, it is believed that infection is acquired early in childhood. The overall prevalence of this organism correlates with socioeconomic status. So the richer the country, the lower the rate of colonization. The poorer the country, the higher the risk, or the higher the prevalence of colonization. Now, transmission is not well known. So many studies have been carried out, but it has not really been well established. However, these are the proposed or the suggested mode of transmission: fecal-oral, oral to oral, or it could be iatrogenic. If a patient comes from an endoscopic procedure and the endoscopes are not well sterilized and reprocessed before they are being used again, it could actually result in transmission of the organism. But like I said, these are all suggestions. What are the virulence factors and how does the organism cause infection? Remember that the gastric mucosa is well protected. The acid produced is quite corrosive, so the mucosa cells are protected by the production of mucus and the flow of gastric content into the intestine, so you don't have a pooling of corrosive liquid in the stomach. So for the organism Helicobacter to be able to establish an infection, it must be able to overcome these protective barriers present in the stomach. So the mechanism that there are different ones that the organism has deployed to overcome the protective factors that we have in the gastrum. Now, one thing I want you to note is that the resultant infection is dependent on the interplay between the organism and the human host. It is possible for somebody to be colonized and throughout his or her life, the person does not come down with any symptoms. However, in some other individuals, they come down with either peptic ulcer disease or they may come down with some form of malignancy. So there's an interplay that is not entirely understood between the organism and the host. But be that as it may, there are certain things that we know about the organism. One is that this organism produces a lot of adhesins that can help it bind to the gastric epithelial cells. Remember that for colonization to occur, the organism must be able to attach to the binding site. So there are different adhesins that this organism produces to effect this. Remember that the content of the gastrum is acidic. So this organism is well prepared for that environment. How does this organism achieve this? It produces the enzyme urease. Now urease will break down urea into carbon dioxide and ammonia. The ammonia will dissolve in water, forming ammonium hydroxide. Ammonium hydroxide is an alkaline agent. So this organism effectively neutralizes the acidic nature of the gastrum. So it attaches, it starts the colonization process, then it starts to neutralize the acidic nature of the gastric environment. Apart from that, the organism is highly motile and it has flagella. These flagella gives it like a drilling motility. So it is able to drill itself through the mucous barrier. So apart from making the environment less acidic, making it alkaline, the organism is actively motile, moving through the mucous barrier. Then it has what we call different pathogenicity island. Remember I told you the last time that the pathogenicity island converts a non-pathogenic organism to a pathogenic organism. Studies have shown that strains of Helicobacter pylori that lack the CAG pathogenicity island are not pathogenic. So the presence of the CAG makes it a pathogenic strain. With CAG, a lot of other molecules are secreted by the organism that will allow it to evade the immune system. Okay, so this could lead to suppression of the inflammatory response, or you can actually have activation of the inflammatory response, and this encourages the adhesion of the organism. So these are the various interplays of virulence factors that the organism deploys to evade the protective mechanisms present in the gastric environment. So like I said earlier, the organism has flagella that it uses to drill through the mucous layer of the stomach. And as it does that, it also affects the pH of the immediate environment through the production of urease. And the effect of this is to make the mucin less gelatinous. So the mucus that is being produced becomes more watery, less viscid. When it is less viscid, the organism can swim faster or can drill its way faster down to the epithelial cells. Now, the various adhesion proteins that are required for the organism to colonize the stomach includes the BAB-A, which stands for the blood group antigen binding adhesins, the SAB-A, which is sialic acid binding adhesin, and the OIPA, the outer inflammatory protein. All these are recognized adhesion proteins associated with pathogenic strains of these organisms. There are other less studied adhesion proteins like the DUPA and the FLA. When we talk of infection, you can have persistence. So the patient can be treated, but the patient is persistently infected. This has been associated with strains that produce the VAC-A toxin. VAC-A stands for vacuolating cytotoxin. It is a proteinaceous cytotoxin, and it induces intracellular vacuolation in cultured cells. It suppresses the T cell response to H. pylori, and it can induce apoptosis in vitro and causes gastric tissue damage in vivo. It has been shown to be a passive urea transporter, thus increasing the permeability of gastric epithelium to urea. Now, everything that I've just told you about this vacuolating toxin is geared towards destroying the epithelial cells. Increased permeability to urea is toxic to the epithelial cells. So this organism allows more urea to get transported to the epithelial cells, destroying them further. It suppresses T cell response. So the organism continues to trigger this inflammatory response while it is suppressing the ability of the immune cells to get rid of it. The CAG-A pathogenicity island, that's what the PAI stands for. The CAG stands for cytotoxin-associated gene. Then the PI is the pathogenicity island. This genetic component is responsible for the production of CAG-A. The CAG-A is necessary for VAC-A expression, and we've already explained what VAC-A does. CAG-A is not cytotoxic, but it is antigenic. So we can actually screen for antibodies towards CAG-A in patient samples. CAG-A is translocated via a type 4 CAG secretion system after bacterial attachment, where it induces morphological changes. Now, type 4 secretory system are systems that allows extracellularly formed components to be translocated into a potentially cell to be infected through a mechanism that is similar to what you have when you use the hypodermic needle. So this is like injecting the CAG into the enterocyte once you have binding. So the expression of this CAG-A gene is strongly associated with peptic ulceration. And studies show a strong evidence between the production of CAG-A and malignant transformation. Now, we've discussed what can happen when the organism colonizes and infects a person. The different virulence antigens and the evasion of immune system we have already discussed. So what could be the outcome, the possible outcome of infection? Remember, I told you that there is an interplay between the organism and the host. So it makes sense that the outcome is variable. So you can't really predict. You can have patients with antral predominant gastritis, and these are the patients that are predisposed to duodenal ulcers. You can have patients with corpus predominant gastritis with multifocal atrophy. And these are the patients that are more likely to develop gastric ulcers, gastric atrophy, intestinal metaplasia, and eventually carcinoma. So the interplay, you don't know where this organism will decide to reside or colonize once the stomach is infected. So here we have the natural history of Helicobacter pylori. And I took this from an article by these two authors. So like we said, there is exposure during childhood. And then you have timeline here with here we have features associated with the infecting strain. You have those that have that result in low level of acid production, and those that result in high level of acid production as a result of the presence of this organism. So what could happen over time? You have the acute infection early in childhood. If the level of production is low, you can have chronic infection. Okay? But as the level of acid production increases, there are different outcomes that could result from chronic infection. You can have a corpus predominant atrophic gastritis, which is a predisposing factor to eventual malignant transformation. You can have non-atrophic pangastritis, which ultimately results in asymptomatic H. pylori infection. Or you can have antral predominant gastritis, which eventually leads to gastric ulcers. So these arrows shows you the different outcomes that could result from a natural infection in a person who was exposed early in life. As you can see, the presentation is quite variable, and you have an interplay between the host and the bacterial agent. So how do you make a diagnosis? The patient, of course, must have presented with symptoms and signs of peptic ulcer disease, okay? So you can have the invasive and the non-invasive approach to diagnosis. And when we decide to evaluate the sample in the laboratory, what are the characteristics that we are interested in to make a diagnosis? Culture is not your first line of management. Culture for this organism is indicated after failure of second-line therapy. What are the samples that you can send in for culture? You can send in gastric biopsy in a transport medium. This is suitable for culture. It's also suitable for histology, and we can also determine the presence of the urease enzyme. You can send in feces. We can perform antigen testing, nucleic acid amplification assays such as PCR, or we can perform culture. You can send in blood. Here we screen for the anti-H. pylori and anti-CAG-A antibodies in the serum. Remember, I told you that the CAG-A is antigenic. When we culture, selective media that could help include Skyros agar, Brucella agar with 5% sheep blood. And then we incubate for 1 week under humid microaerophilic conditions. Remember, I told you that the microaerophilic condition is a well-defined condition. So you have 10% CO2, 3 to 5% oxygen, and 85% nitrogen in an anaerobic jar. You do not require a catalyst to make it a completely anaerobic environment because the organism is a microaerophile. It still requires little oxygen, so you don't incubate anaerobically. That's why we don't use a catalyst. So the organism is spiral shaped and it's gram negative. It is highly motile. It is microaerophilic and it takes a while to grow. The biochemical tests that we usually use to identify the organism include urease test, catalase, and oxidase test. The organism is urease positive, catalase positive, and oxidase positive. Most strains are susceptible to cefalotin and they are resistant to nalidixic acid. So if you're told that you have a gram-negative organism that is urease positive, catalase positive, oxidase positive, and susceptible to cefalotin, or resistant to nalidixic acid, and the organism was isolated under microaerophilic condition, you should be thinking of H. pylori. In terms of clinical diagnosis, there are other approaches that you can use. You can perform endoscopy. You can perform the CLO, which stands for Campylobacter-like organism test, which is a rapid urease test performed in the clinic. Or you can use the carbon urea breath test, in which you use radioactive carbon. So what's the guide to management? I'm not going to go into details of the drugs because I'm sure you'll have discussed this in your pharmacology lectures. What you should note is that it's a combination of an anti-secretory agent and different antibiotics. So in conclusion, understanding of the pathogenesis of H. pylori continues to evolve, and you should note that factors involved involves both the host and bacterial factors. Infected patients are usually asymptomatic, so you can have variable presentation, and the organism is strongly associated with peptic ulcer disease and gastric cancer. It is a type 1 carcinogen. Making a diagnosis could be clinical or laboratory based. It depends on you, the managing physician. Treatment involves the eradication of the organism and the reduction of gastric acid production. Any questions from the two lectures? Anything in the chat box, Doctor?
No, doctor, there is no questions in the chat.
Okay, so have a good day, everyone.
Thank you, doctor. Have a good
Bye.
day.
Thank you. Bye.